All right, Wednesday, March 18th. This is going to actually be most likely the shortest video of the week. There's not a whole bunch I want to do new with you today. I just want to show you how to do a 16 square Punnett square, which is a dihybrid cross. Some of you probably even done that before in the past. And I wanted to briefly say something about meiosis again, because I didn't really fully touch on it as much as I would have wanted to. And hopefully we can get back to school and do the meiosis activity that I've created. But if we look at your mom's chromosomes, these are just two pairs of the entire 23 pairs. They all range, there's tons and tons and tons of genes on each of the chromosomes. But if you look at your mom's chromosomes, she has one chromosome that she got from her mom, your grandmother, and one chromosome that she got from her dad, your grandfather, on your mom's side. And those are full sets of DNA that she got from each of her parents. And she has, again, 46 of these chromosomes, 23 from each parent. However, they do switch around during the process of meiosis. And if you remember, meiosis is when the sperm and the egg cells are being created by either the mother or the father, respectively. What happens first, it's all very similar to mitosis. The stages are the same. The stages just happen kind of twice so that there's an extra round of division to make half of the DNA as the original cell had. We've talked about that many times before. But what happens is the same thing with replication of DNA. The chromosomes are duplicated. So here's the duplicated one from your grandfather, the duplicated one from your grandmother inside your mom's cells as she's preparing to prepare her egg cells for reproduction. And again, these are just small examples. Really, there's 23 of these going on, and they have millions of genes on them. But when they line up here at prophase of the first step of meiosis, what happens is genes will switch back and forth, called crossing over from the chromosomes that your mom got from her dad and her mom. And so what that ends up giving us, by the time they separate out and go into four different possibilities of cells that can be the egg cell, you might get something like this. And this is just one possibility. She has genes that she rearranged to prepare her egg cell, and they have genes from her mom and her dad. So both of your grandparents on your mom's side are going to have genes represented in the egg cell that is eventually going to become you. And now this will happen completely randomly, completely differently, with all types of chromosomes rearranging in all different ways along the metaphase plate and crossing over, switching all kinds of genes every time she prepares an egg cell, which is why you and your siblings are different combinations of genes from that your mom gave to your siblings and your dad gave to your siblings. The exact same thing is going to happen in your dad's cells as he's preparing a sperm cell. And so here would be his, with different resulting combinations that he got from his father, your grandfather, and his mother, your grandmother, on your dad's side. And when these combine, these are going to be your sets of DNA that you got from all different of your four grandparents. You'll be big R, little r, you'll be two big H's, you'll be big B, little b, and you'll be big G, little g. So you're going to be hybrid for everything except the H. But if you notice, you're going to have DNA that is from all four of your grandparents. And that's how you get these unique combinations to create an individual. And these are where we get, the alleles to put on Punnett squares to try to figure out what the possibilities of what you could have might be. But anyway, so that's meiosis. I hope we can do more in class with it. But I wanted to show you how to do a dihybrid cross. And this is when two traits are being considered instead of just one. So far, we've only looked at one trait, but sometimes you might have two traits that are going to need to be considered. So let's say we have a plant. I'll do the easiest example with a plant where we're going to look at height and color, okay? And let's say that T is tall and that's dominant to little t, which is short. And for color, let's say that the dominant color is G, green. And if it's little g and you're recessive, you would be yellow. So let's go with that. If we mate two parents, let's say the first one is hybrid for both. So that's tall and green, but hybrid for both. That can be the father. And we're going to cross that father plant with a short mother, who's also still hybrid for the green color. So let's do one like this. There's all different kind of combinations you can have, and tonight's problem set is all on these 16 square Punnett squares that you can try with all different types of combinations. But to line these up on the mother and father side of the Punnett square, you kind of have to do what I think of as like the distributive property in math. This t needs to go with this g, and this t needs to go with just g, and then this t needs to go with this g, and this t needs to go with this g for one side. So basically it would look like TG capital, then big T little g, then little t big G, then little t little g. Okay, and so that's that first parent, the father. Same thing for the second. It's going to be all little t's, but you have to use each one of them because that's how the combinations would work during meiosis as you're splitting up these alleles. You'll have little t big G, little t little g, little t big G again for the second t allele that's a possibility, and then little t little g again. And so that's the setup. And usually once you get to that point, it's much easier. But then you just fill it in. So this one's going to be big T, big G, big G, big T, big G, little g. You can fast forward through this if you don't want to watch me do all of these, because I'm sure you understand. So go ahead and fast forward. And then we're going to look at the combinations. Oops. Little t, big G, little t, big G, little g. And hopefully you all understand how I'm doing it. Okay. So here we have it all filled out, and now you just kind of had to see what the probabilities are out of 16, and that gives you sort of a proportion or a percentage that it's going to be. So I have, if I'm going to look at any that are tall and green, that's going to be here, tall and green, 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 here's tall and green, and that's it. 1, 2, 3, 4, 5, 6. So we have 6 out of 16 chance tall and green. Okay, let's look at tall and yellow. We have tall and yellow 1, 2, just 2. So we have 2 out of 16 tall and yellow. And you'll notice these fractions are going to simplify. You could put those out of 8. So tall and yellow. Next, let's look at short and green. We have 1, 2, 3, 4, 5, 6. 6 out of 16 short and green. And I guess that's going to leave, what, 2 of them that are short and yellow? And so you can calculate those proportions. This would really be 1/8. 2 out of 16 is 1/8. And we have 3/8 for the 6 out of 16. And so you add those all up and it's just proportions out of 8% chance. And you can make those as a decimal, but these are the proportions of the phenotypes. And simplify your fractions, put them out of 8. But I'll accept fractions just for this. Usually we like decimals, but for this you can leave it as fractions. But do simplify them. Don't put them out of 16 if they're even numbers like that. But that's how you do a dihybrid cross. You might have different parents that you will mate and mix up, and you have different proportions and different probabilities. But dihybrid crosses are when you kind of look at 2 different genes at the same time. So that's all for today. I don't want to overwhelm you with too much. If you are not learning enough from these videos, then YouTube some other videos, because this stuff is all over the internet with how to do these types of genetics problems and Punnett squares and crosses of some sort. Next week is going to be a lot harder for us, but this week is pretty simple stuff. Yesterday we looked at the basic Punnett squares and how to calculate probabilities, the exceptions to the main rule where the hybrid might be codominant, where they show both traits, or incomplete dominant, where it's a mixture of the two. And we also looked at multiple alleles, more than just two different letters, like the blood types as an example. Hair color is another example of that type. And you maybe hopefully know a little more about meiosis now. I'm not gonna go on. I might post Thursday's video though today at some point early if I can get around to it. But that's all that you need to do for Wednesday. The problem set for these 16 square dihybrid crosses is up for you also. So you can begin those and do those within 24 hours. And you can also look at the answers now to yesterday's work that is up as well. So you can look at the answers to that and make sure you understand how to do it. Although I'm giving you the answers, I still want to see that you've been doing them. So please keep doing all of the official problem sets labeled with a date on some sort of sheet of paper. You can run them all together, that's fine. But just show me that you've done your problem sets by writing them down. I will have you send them to me somehow once the week is over. But just keep doing them each night within 24 hours so that you're keeping up with what we're doing. Again, this should be easy for everyone. Most people are probably like bored by watching these videos because you know how to do this already, especially if you take genetics. But even in 9th grade ST Bio, I'm sure you learned most of this. But as always, feel free to email me with any questions you have. We're going to continue until we get any updates from the school system. We'll finish the easy genetic stuff this week and then do some hard stuff that I know you've never done, some college level genetics next week. So next week's going to get a lot harder. Again, the Mastering Bio is long, it's extensive. And it can be a little bit challenging. Refer to your textbook. It's on chapter 14. Mostly all of it is from chapter 14. There might be pieces from chapter 15, which is tomorrow's part, where we are going to talk about sex-linked genes, things that are on the X or Y chromosome and have to do with what gender you are. But tune into that video once you're done and you're good with these 16 square dihybrid crosses, and have a good evening. Hope you guys are doing well, and email me with any questions that you have.